The human musculoskeletal system is not a static structure. It is a highly dynamic, constantly remodeling biological engine. However, as an individual crosses the physiological threshold of 65 years, this dynamic equilibrium undergoes a catastrophic shift. The rate of muscle protein degradation begins to aggressively outpace the rate of muscle protein synthesis, leading to a relentless, involuntary loss of skeletal muscle mass and strength. This condition, clinically defined as sarcopenia, is arguably the single greatest threat to the functional independence, metabolic stability, and longevity of the aging population. For decades, the medical consensus treated this wasting away as an unavoidable consequence of chronological aging, a natural slowing down. Modern cellular biology has unequivocally dismantled this defeatist paradigm. Sarcopenia is not an inevitability. It is the physical manifestation of a profound metabolic communication breakdown known as anabolic resistance. In a 65-year-old body, the muscle cells essentially become deaf to the standard nutritional signals that previously triggered growth and repair in youth. You can consume a healthy diet, but if you do not provide a massive, targeted, and concentrated biochemical signal, your body will refuse to assemble new myofibrillar tissue. Instead, it will cannibalize its own skeletal muscle to harvest amino acids for vital organ function, leaving your limbs frail, your balance compromised, and your metabolism sluggish. To halt this silent internal decay and actively force the aging body to rebuild its structural foundation, one must execute a highly specific dietary intervention, utilizing 3 precise biological agents. To understand the necessity of these 3 foods, we must first examine the microscopic signaling pathway known as the mammalian target of rapamycin, or mTOR. This enzyme complex is the absolute master regulatory switch for muscle protein synthesis. In a young body, the mTOR switch is highly sensitive. A simple bowl of oatmeal or a light walk is enough to flip it on. But in an aging body suffering from systemic low-grade inflammation and oxidative stress, this switch becomes rigid and resistant. To force it into the on position, the body requires a massive, concentrated surge of a specific essential amino acid, leucine. Most seniors fail to build muscle because they engage in protein grazing, eating small amounts of protein throughout the day. This never produces a spike in blood amino acids high enough to cross the leucine threshold, meaning the mTOR pathway remains completely dormant. To break this resistance, the first food in the sarcopenia defense protocol must be a highly bioavailable, leucine-dense protein matrix, specifically whole eggs combined with a high-quality whey protein isolate. When you consume a substantial bolus of this protein combination, aiming for roughly 35 to 40 grams in a single sitting, you flood the portal vein with a high-velocity surge of amino acids. This sudden, massive concentration of leucine physically binds to the cestrin-2 sensor proteins within the muscle cells, aggressively releasing the brakes on the mTOR complex. This is the chemical command that forces the ribosomes to begin assembling new muscle fibers. The whole egg is critical here, not just for its flawless amino acid profile, but for the dense lipid emulsion provided by the liquid yolk. The yolk contains essential phospholipids and cholesterol, which are the foundational sterile backbones required for the endogenous synthesis of all anabolic hormones, particularly testosterone and dehydroepiandrosterone. These hormones are notoriously low in the aging demographic. By utilizing eggs and whey isolate, you provide both the raw building blocks and the hormonal raw materials necessary to initiate structural repair. However, delivering the amino acids to the cell and flipping the mTOR switch on is only the first step in the complex biological assembly line of muscle hypertrophy. The cell has received the signal to build, but it requires an architect to read the genetic blueprints. In human physiology, that architect is active vitamin A, specifically functioning as retinoic acid. Inside every muscle cell, located directly on the nuclear envelope protecting your DNA, are retinoic acid receptors. When active vitamin A binds to these highly specific receptors, It acts as a transcription factor, sending a powerful signal to the DNA to initiate the actual transcription of the genes responsible for creating myosin and actin, the contractile proteins that physically comprise the bulk of a muscle fiber. Without adequate retinoic acid, the cellular machinery is blind. It cannot execute the construction commanded by the leucine spike. This brings us to the second, highly unconventional, yet medically essential food in the protocol: thermally processed, heavily boiled carrots. Carrots are a profound botanical reservoir of beta-carotene, a provitamin that the human liver converts into active retinol. Yet, the medical efficacy of the carrot is entirely dictated by its physical state. In a raw carrot, the beta-carotene is locked behind a formidable matrix of indigestible cellulose. The aging human digestive tract cannot extract it, resulting in an absorption rate of merely 3%. To weaponize the carrot for skeletal muscle growth, it must be boiled aggressively. Thermal processing physically melts the crystalline structure and degrades the cellulose walls. This releases the beta-carotene in a highly bioavailable, free form, increasing its absorption by up to 600%. Crucially, because beta-carotene is strictly fat-soluble, consuming these boiled carrots alongside lipid-rich egg yolks creates a perfect transport mechanism. The fats emulsify the carotenoids into microscopic chylomicrons, shuttling them across the intestinal barrier. They move directly to the liver for conversion, ensuring your muscle cells are flooded with the exact transcriptional ink required. This writes the genetic instructions for new muscle tissue, promoting skeletal longevity and growth. The third and final component of the Sarcopenia defense protocol addresses the bioenergetics capacity of the aging muscle cell. Building new tissue requires a massive amount of cellular energy in the form of ATP. Furthermore, to signal the body to retain muscle, the muscles must be placed under mechanical tension. They must be used. However, older adults frequently suffer from a severe depletion of intramuscular creatine stores. Creatine phosphate is the primary substrate used to rapidly regenerate ATP during explosive, high-tension movements, such as standing up from a low chair or catching yourself during a stumble. When creatine stores are depleted, the muscles fatigue prematurely, and the signal for growth is weak. The third food intervention is the strategic inclusion of high-quality, lean red meat, such as grass-fed beef, or, for a more concentrated intervention, exogenous creatine monohydrate supplementation. By replenishing the intramuscular creatine pool, you restore the explosive contractile power of the type 2 fast-twitch muscle fibers. These specific fibers are the most critical for maintaining balance and preventing catastrophic hip-fracturing falls. The implementation of this muscle-building triad is not a loose dietary suggestion. It must be executed as a strict clinical protocol. The aging body must break out of its overnight catabolic state, where it breaks down muscle for energy, as rapidly as possible. Therefore, the protein bolus, the boiled carrots, and the creatine source must be consumed together as the first primary meal of the day. This creates a massive synergistic anabolic window. The lipids from the eggs facilitate vitamin A absorption, while the leucine surge from the whey triggers the mTOR pathway. And the creatine ensures the cells have the energetic capacity to execute the repair protocols. However, we must address the silent epidemic plaguing the over 65 demographic and threatening to derail this process: hypochlorhydria, or low stomach acid. The parietal cells in the aging stomach often fail to produce enough hydrochloric acid to denature complex proteins or activate pepsin, the enzyme that cleaves proteins into absorbable amino acids. If you consume 40 grams of protein but lack the stomach acid to digest it, the protein will putrefy in the gut, causing severe bloating and providing zero anabolic benefit to your muscles. To guarantee the success of this protocol, you must chemically support the digestive environment. Consuming a tablespoon of raw, unpasteurized apple cider vinegar, diluted in a small glass of water 10 minutes before this anabolic meal, safely lowers the gastric pH. This mimics the acidic environment of a youthful stomach. ensuring the heavy payload of protein is successfully dismantled and absorbed into the portal vein. When this highly specific, targeted nutritional protocol is maintained with uncompromising consistency over a 30-day cycle, the internal physiological shift is nothing short of a systemic metamorphosis. The initial changes manifest in the realm of neuromuscular efficiency and metabolic stability. The pervasive sensation of frailty, the heaviness in the limbs, and the exhaustion from simple tasks begins to evaporate. As the mTOR pathway is repeatedly and forcefully activated, the muscle cells regain their youthful capacity to uptake nutrients. The mitochondria within the muscle fibers become denser and more efficient. By the end of the month, the macroscopic results become clinically evident. You will observe a measurable increase in grip strength, a crucial biomarker inversely correlated with all-cause mortality in the elderly. The ability to rise from a seated position without using the arms and the restoration of a confident, stable gait confirm that the accretion of new myofibrillar tissue has occurred. Furthermore, this newly built muscle acts as a massive metabolic protector. Skeletal muscle is the body's primary sink for disposing of circulating blood glucose. By rebuilding your muscle mass, you exponentially increase the number of insulin receptors available, drastically improving whole-body insulin sensitivity and aggressively defending against type 2 diabetes. You are essentially shifting your body from a fragile, wasting trajectory into a robust, building trajectory. To accept the loss of muscle after age 65 as a normal, natural part of aging is a dangerous medical fallacy. Sarcopenia is a reversible metabolic emergency. By understanding the biochemistry of anabolic resistance, the necessity of the leucine threshold, the transcriptional power of retinoic acid, and the bioenergetics of creatine, you possess biological authority. This protocol proves that the resilience of your later years is not dictated by the passage of time, but by the exact chemical information you demand your cells to process, ensuring your muscles remain powerful and fully capable of supporting a long, vibrant, and fiercely independent life.